Welcome to your first flipped lesson in science. The title of this flipped lesson is called Scientific Method. All the notes for this flipped lesson and pretty much every flipped lesson you do in science will be taken in your vocab notebook. That's your spiral notebook that you have dedicated for your science vocab notebook. So everything on the boards that you see you're going to write down, and that way you have a complete set of notes that we can use as application in the classroom. All right, you're given class time for this first flip lesson to watch it and get the notes down. Just know that you're not always given this class time, that sometimes a flip lesson has to be done at home. So first of all, we're going to start with the definition of the scientific method. First of all, you have to understand that this is a process, all right? And what it does, it identifies a problem, it makes a guess about the problem, you collect data about that problem, you analyze and look at all the data that you have, and then you make a conclusion about what you identified as being a problem. And it's a very cut and dry set of steps, and we're going to practice with this the first 3 weeks of school. So down here we have step one, which is define the problem. All right, what problem do you have? First of all, you have to be clear. All right, you're identifying one specific problem. You identify the problem in a form of a question, and like I said, you keep it to one idea, because you don't know if it's the one idea you've created that is causing the problem if you try to throw other things into this idea as well. Step two, once you have your problem, you need to collect information. You use the information that you collect to make a guess. Well, where do you get the information from? It could come from a textbook or text that you read, a reference book, online, other people, observations, just background knowledge that you may have about a topic. If you don't know much about the topic, that's where you would go to some outside resources to use as a basis for your collecting of information. You need to read and think carefully about the information you collect in these different sources. Once you've collected some background information, you want to formulate a hypothesis. And basically your hypothesis is an educated guess. What do I believe is the answer to this particular problem? And it needs to be something logical and educated, not just some random thought. You'll use the information that you've collected up here in step two to make an educated guess, or your hypothesis. Once you make your educated guess, the next logical steps is you need to test your hypothesis. This is the word that you would know as experiment, okay? And in every good experiment, there must be three parts. Part number one is the independent variable. What are you going to test? So let's say that you were trying to decide if adding vinegar to plants would make them grow better. What you are testing is adding vinegar to your water, okay? You're testing, does vinegar help my plants grow? The next piece you have to have in a good experiment is called a dependent variable. This is what you are going to measure. So if you water your plants with regular water and water the other set of plants with same kind of plants but different pots with water that has vinegar, what you would measure with these two different groups would be, do the plants grow, and if so, by how much? So that's your dependent variable. And then you always have a control. The object that you use is a comparison. So if we think about that experiment where you have a pot of a sunflower and another pot of a sunflower, one of them you water with regular water, one of them you water with water that has vinegar added to it. What you are testing is, does vinegar affect how my plants grow? All right, what are you measuring? How much do the plants grow? Which of those two pots is your control group? That control group is the pot that you didn't do anything different to, and that would be the pot that just had the water added to it, not the water and the vinegar. And if you were to graph your results, always your independent or your dependent variable goes on this axis known as the y-axis. Your independent variable goes on this axis down here. It is called the x-axis. And finally, your last step is to draw conclusions. Now, this is not draw a picture, all right? This is actually come to a conclusion. And you shouldn't just jump to conclusions. Don't think halfway through an experiment, oh, I know the answer. Fulfill the experiment, make sure it's a solid experiment, and don't jump to conclusions. Sometimes thinking simple is usually the right way to go. All right? Trying to make it too complex and too hard is going to make things confusing. So keep it simple. And you use the data you collected during the experiment to support the conclusion you came up with. All right, and it's always important to say yes or no is your answer. So if we think about the pots, Does adding vinegar to your water help your plants grow more than just regular water? That is a yes or no answer. There are no maybes in science, okay? And you can stick with yes or no, which means you can never be wrong. It's either yes or it's no, and you could defend both sides as long as you use data to support your answer. Now our plan is to take this information from the scientific method, and there will be a link on my website where you will conduct an experiment online about crickets and what things affect how fast a cricket will chirp out in nature.